All right. Let's talk about what we're actually going to do this year. What do the data we'll collect look like? And by the way, 3410 students, you've already seen some of this. And so I'm going to go through it a little fast, but just to remind you. And the other thing is, this comes from my article about this. So another way for you to learn about SPR will be to look at that article, because it talks about using this exact instrument. It has a picture of it. So this is some of the things from that article. Remember that you're putting different concentrations through, and you're measuring how high the numbers go. And so you can measure at this point right here. This point is where it's the flattest, you're the closest to equilibrium, and you measure just the height of that point, and you do a plot of the height versus the concentration. And the height versus the concentration will give you the KD when you do the fit to this curve will give you the equilibrium constant. Again, this is a chapter 4 thing, but the one thing is you can measure how much is binding at the point right before it drops off. That is equilibrium measurement of binding. And if you look at different things, like here's a case where we had wild-type protein. These are mutants that bound worse, and they have lower responses. You see that this one's lower than that one. These are all on the same scale. If a mutant binds better, we actually have higher responses, and you see how you can actually see it curving up. It's actually hitting sort of an asymptote, and you can see that it's curved. And so you can see from how the line curves, if it's pretty much straight, it's low affinity, but if it curves, it's high affinity, and the equation will give you the equilibrium constant of binding. The other thing you can do is you can look not at this point, but at how the line curves up and down. This is chapter 6 and 7 material. This is kinetics. But you can see right here, you see how this line's curving down faster than this one. This one is more stable in its off rate because it's falling off more slowly. It's stickier. So you have the injection here, on rate, off rate. And you can just see from comparing these, this one looks like it's slightly stickier than the other one, but they're pretty much the same. And what this is showing is that these are actually, these are two very different proteins, but it's showing that this one's probably slightly tighter binding than the one on the left. And so just look at how it curves. How far up does it go? How's it curving on and off? This is what I'm hoping you will see. I'm not sure if you'll see something closer to this. We'll deal with it, whatever it is. But you're going to put a fragment on the chip. Here was a fragment that was put on the chip called fragment B. And then we put an antibody over the chip that would bind to that. And so the B on the chip, the anti-B antibody, bound, and it caused it to go up. And you see that there's binding, and then it stayed bound when it was all the way up. So the B on the chip plus the anti-B antibody resulted in the elevation of the binding. This binds really well. On the other hand, if we put the antibody on the chip, we also see binding, but we see much lower binding. You see how this response is much lower, noise is big. In fact, it's hard to even see this. And so we have to talk about what to do about data like this. But the thing is, this is barely working, but it is still working. And it's showing us that we get about the same response. We're actually getting a similar result from this. Hopefully your data will be closer to this than to that, but we will have to see. That's how it will. We'll have to see how it works. So I'm not going to spend too much time talking about the previous SPR data that we collected. There's 1 year, so this was in 2 years ago. This is when we had students designing different fragments. These are region B fragments, and they bound they bound antibodies in different ways. I'm not going to go through it except to say this is what antibody binding looks like. This is what hopefully we'll see something like this when we throw our antibodies onto the chip. And this is cool because this is the case where the student designed a bigger fragment and we put the antibody on the chip. So you remember how we got a really low signal for just a regular size fragment. This is the antibody on the chip, a bigger fragment, bigger signal. And the bigger signal meant that we could actually measure both an early and late time points for it. And I'm convinced that that's better, much better than what we had before. Okay? Now we're getting to what we have done in recent years. This is where we used whole serum. The blue lines are where we had a MG281 protein. It's a different protein that the MG makes. We put that on the chip. We put the whole serum on. We put the whole serum on. We saw binding. The lines went up. Then we depleted the antibodies out from it. We still saw binding, but we saw less binding. And it was very repeatable. Even though it was a small effect, it was repeatable. And so we actually said, we think, number one, that there's antibodies in the serum that bind. We can take them out. Number two, there's still something in the serum that's binding. That's not IgG antibodies. We found out actually what that was. We think it's alpha-2 macroglobulin. Long story. But basically that is what we want. And the main thing about that, we were able to use serum. The other thing that we've done recently, we've actually used sheep red blood cells. And so that's some of what we want to do is our collaborator needs us to do a few more experiments. And so the idea is using whole red blood cells, and they're sheep. So it's like handling meat basically, but it will be whole red blood cells, and it's part of our project. I'm not sure how much of our project it will be. We'll have to see how the fragment stuff is working, and we'll be mixing it in. So you might talk to me if you have a reason for wanting to go one way or the other. But otherwise, then I'll just give you all different things to do. We're going to try our fragments that we're purifying. We're going to put them on the chips. We're going to measure the antibody binding curves. Hopefully some will look like that type binding we saw, but the more mutated fragments will hopefully bind less. That's our idea, that the antibodies, the alanines, will bind antibodies less. And we're also going to start with, we have CM5 coupled chips with the other half of the MGP dimer. We have the MGP-C, but these are the ones that we know bind to sheep red blood cells. And so this is what I would be giving, and like if this doesn't work, okay, then we'll move on to doing this. That we have multiple projects, and you don't always know what's going to work in the lab. But I want to let you know this is coming down the pike, and you might be doing some of this if that's what, if working in the lab with that is what you can do. I'm hoping that we will see binding, but it will be weak binding. And it's always a trick when we're dealing with weak binding, dealing with fluctuating baselines and stuff like that. But we will basically just keep working on it, and we just don't expect research results to be perfect. That's part of the whole lesson, that we'll work through it either way. Deb? What's that? Why didn't you choose sheep? Because they have the same sugars on them, and they're similar to human, but they're not human. I don't want to work with, I'd rather work with sheep blood than human blood, basically. There are a number of different species that we could use, but I think that the sheep blood is what they decided on. So, but I do want to let you know that it is an animal product. It is going to be actually, it's going to be blood in some ways, and but we've gotten it to work, and it's really, it shows how whole cells bind to the thing. So it's actually valuable and helpful data. But we do have two projects. And I just want to let you know, I didn't know about this, that this project would need to be moved up in timeline until Friday. So I just found out. But I want to try to get some of this done too. Okay. Don't worry if the binding data is too low to measure. Objective one is to get you to try. Worst comes to worst, I have my ways to give you stuff to learn on, data to learn on in another case, if we have an experiment that just doesn't work. But that's sort of the risk that we have to take. If we're doing something no one's done before, there is the slight chance that it won't work at all. But, you know, I mean, we aren't even talking about the results of the Ericsson. We're just saying we will try it. So let's try it and let's see what happens. That's the talk. So I know it's a lot, but that is